So the protein that I was assigned is lysozyme, and this article I came across through the school's library database is just called lysozyme, and the authors of it are Elliot F. Osserman, Robert E. Canfield, and Sherman Bayshok. All the work done in the article was conducted in London, with other tidbits pulled from prior experiments or researches done elsewhere, like Paris. Some of the things I found interesting in this article, if I can go to it real quick, is something about, right here, mammalian lysozymes. So lysozymes, as far as like research goes, researchers are really using or back then, I don't know about now, but researchers were using hen eggs and chicken eggs because in the egg whites, lysozymes are readily available in large quantities. But let me just read this all in the yellow. Fleming described the presence of lysozyme activity in a number of mammalian tissues, but the purification of large quantities of enzyme was less readily achieved than that of hen egg white lysozyme. Then in 1966, Osserman made the important observation that patients with myelomonocytic leukemia often excrete large quantities of this enzyme in their urine, with some patients excreting in excess of 1 gram per day. I found that to be interesting. Something else which goes a little bit further into the leukemia thing is right here, this first one. As noted earlier, the primary structure of human leukemia lysozyme is remarkably homologous with that of hen egg white lysozyme. The next one down here. While we continue to designate our material as a leukemia lysozyme to identify its source, it is apparent from amino acid sequence studies by Joels and colleagues that leukemia protein is identical to the lysozyme in human breast milk. I found that to be interesting since it's like very, very similar, if not the same. And the last thing that I came across that I found interesting is right here. This is when they were conducting research into 3 black swans with their egg white lysozyme. A totally unexpected observation was made during studies on individual egg whites from 3 black swans. It appeared that this species contained not 1 but 2 lysozyme in its egg whites. One of them was immunologically an Ebden goose type, whereas the other one was immunologically a chicken type. So they found 2 different types of lysozymes in the goose egg whites, and I found that to be interesting. And that is all for now.